Okay guys, so we are done with chapter 3. We are going to go on to chapter 4. Just to give you an update on our time schedule. After chapter 4, we're going to have a big unit test. So start looking over your past material during this one as well. Reason being, guys, at the end of this, the unit test is going to be chapters 1 through 4. So you'll have to know pretty much like the basic stuff, the overall like outline of chapters 1 through 4 for your big test. But anyway, we're going to get into the cell for this one. And it's just going to be like a basic overview of the cell, guys. We're not going to go crazy in depth because later on, we're going to take each one of our organelles, take each one of our different parts of the cell, and break them apart in individual chapters. Okay, so you guys already know that the cell is the smallest unit of life. It's the smallest thing that is considered living, okay? Anything smaller than the cell is not living, and that's why we say organelles are not living, okay? Okay, so here is a picture of a brain cell, guys, and I know these look very different than your regular cells. Just so you guys know, cells differentiate. So when you are very, very, very young, still being developed in the womb, your cells start to differentiate. We have different stem cells, and then they start to get different functions. So we have brain cells, lung cells, all these different cells. The brain cell, they're very just unique just because they have all these different parts that latch on to other brain cells to convey those messages. So that way, your body can respond to however you want it to respond. So our cells are very tiny, guys. They are between 1 and 100. That little U guy there is micro. So they are micrometer, okay? Very small. And obviously prokaryotic cells are going to be much smaller than eukaryotic cells. Usually they're about 10 times smaller, okay? So prokaryotic cells, much smaller than eukaryotic cells. Your book then goes into some different microscopes that we can use to look at cells and different things, not just cells. But the first one is the light microscope. These are the ones you guys have used in the classroom before. They're good. We need light to be able to pass through the organism to be able to see it. So we need to make sure that the organism is very, very, very thin. All right, that might even be like cutting it. You might have to cut something in order to see it under a light microscope. Light microscopes can go up to 1,000 times with respect to magnification, so we can zoom in 1,000 times on it, okay, which is pretty good. So there's a difference between magnification and resolution. So magnification, that's zooming in on something. If we zoom in on something and it's really blurry, we can't really see it, what's going on with it, that's going to be your resolution. So a little bit different than magnification. Right? And in contrast, guys, pretty easy if you've ever taken a picture before, any kind of photography stuff. With contrast, you're going to put your subject on a background that isn't going to be the same color as the subject. All right. Why that is so you get you're able to differentiate between the two. You're able to differentiate between whatever person you're putting and the background. That way the colors aren't blending in. All right. That's contrast. With contrast, guys, usually what we'll do to help that out, especially with light microscopes, is we might stain or dye our specimen. That way it's not clear, it's not see-through, just like the glass, all right? The glass is see-through that we put the specimen on. So we'll dye it with some kind of dye, and then we're able to see it better. So here is a picture of what we can see with the light microscope there. So you can see between 1 and 100 micrometers. That's what we can see with our light microscope. That's our plant cells and everything like that. Okay. Anything above that we can see with the unaided eye. We don't really need a light microscope unless we're going to want to zoom in. And then we'll talk about these ones later on with the electron microscope. All right, so we already talked about this, guys. We're able to see 1,000 times, zoom in 1,000 times. We're also able to stain things, like I said. Usually the easiest, cheapest one to use is iodine. Okay, we use iodine to stain things. Gives it like usually a brown or a purple or a blue color, but they work pretty well. Okay, so the electron microscopes, there's two main ones that we're going to talk about, the scanning electron microscope and the transmission one. They're abbreviated SEM and TEM. So the difference between them, guys, is the scanning electron microscope, what it's going to do is it's going to shoot electrons to the surface of our specimen. And then what's going to happen there is those electrons are going to hit the surface and get bounced or refracted. So when that happens, we're able to see how far away from the point where the electrons are being fired out to that specimen. And then what happens is it paints a picture based on how far away it was. So the electron is really, really, really tiny, guys, and they're firing out millions of electrons, these electron microscopes. What's nice about that is since they're so tiny, we're able to get a really, really, really good picture of our specimen, and we're able to get all the surfaces because it does like every single bit of its surface and paints a picture based on that. The transmission one's a little bit different. We're going to send the beam through our specimen, okay? And what that's able to do is it's allow us to see the internal parts of that organism. Here's some pictures with a light microscope. All right, cool. Nothing too crazy there. You guys can see we're zoomed in. Some of them we're just going to stain. You can see even some of them up here unstained. So you can see the difference. There's no contrast there. If we stain it, we get pretty good contrast. Here is still a light microscope, but instead they use fluorescent dyes in order to see the specimens, but you can see the difference between them, right? These ones are kind of blah. These ones are a lot nicer, a lot easier to see the different things. And what we're going to do is we're going to use dyes that attach to specific elements. That way we can see what's going on, and that's why we can highlight different parts. Now these ones here, these are taken by a electron microscope. And you can see the surface on the left one here, just like I was talking about with the electrons, guys. We're able to see the surface really well. The electrons go off of each of these little cilia here, and it does paint almost a three-dimensional picture. So the scanning electron microscope gives us more of a 3D representation of what's going on. The transmission one, you could see it's more 2D, it's more flat. That's because we're looking at the internal structures instead of the external. Here's a short video, guys, on pictures taken with the electron microscopes. I think this is really cool to look at. Be able to see a few normal things. This one's gross looking. show you guys a few more and then I'm going to stop. We're not going to watch the whole thing. Okay, so all of these pictures, guys, were taken within a scanning electron microscope. Okay, so we all got the outside picture of it. All right, it was an external picture, so that's how you know it's scanning. The big thing with electron microscopes, guys, is they can go. We said the light microscopes, they're like 1,000 times magnification. Okay, these ones can go like 10,000 times for magnification. So a lot more compared to the light microscope. All right, adding a little zero at the end of 1,000 to get 10,000, that actually makes a gigantic difference there. We already talked about fluorescent markers, guys. We already talked about dyes we can put in there. I showed you a picture using fluorescence. We're not going to dwell on that. So cell fractionation. What this is breaking up the cell into different parts. All right, it might be the organelles. We could even go smaller than the organelles. But what it's going to do is it's going to take things and put them based on their size. All right, some organelles are going to be bigger than others. And I got a little video for you here on cell fractionation.
The cell wall of a bacterium encloses a riot of diverse chemical activity and molecules. To study the components of these cells, a researcher may want to break open the cells and purify the different molecules and structures. One useful method for breaking apart cells is sonication. In this method, a probe emits high-frequency sound waves, which fracture the cells. The contents of the cells spill out into the medium, creating a complex suspension of molecules and fragments. The mixture includes unbroken cells, pieces of cell walls, cell membranes, and proteins such as enzymes. There are two common centrifugation strategies for separating the bacterial components. One is called differential centrifugation. In differential centrifugation, a tube is filled sonicated bacterial suspension and placed in a centrifuge. A second tube contains liquid for balancing the centrifuge during its spin. The first spin is usually set at a low rotation speed, producing a centrifugal force of about 15,000 times the force of gravity. During this short, low-speed spin, the largest and densest materials sediment first, forming a pellet at the bottom of the tube. The pellet contains primarily unbroken cells, flagella, and cell walls. The remaining liquid, called the supernatant, contains smaller and less dense structures. The components of the supernatant can be separated further by a second spin at a high speed, this time providing a force of 100,000 to 150,000 times the force of gravity for 60 minutes. Again, the materials with the greatest size and density move fastest to the bottom of the tube. Ribosomes and membranes sediment under these conditions. Soluble enzymes do not. Instead, they remain in the supernatant. Although differential centrifugation can quickly fractionate a bacterial suspension, the fractions are not pure. Another method, buoyant density centrifugation, provides greater purity. The suspension is centrifuged through a density gradient to separate the components. The tube in this example contains a sucrose solution with a high density at the bottom and a lower density at the top. The cell suspension is added to the top of the sucrose gradient. During centrifugation, the components move fastest down the tube. If a component reaches a sucrose density equal to its own density, it travels no further. The bands closest to the bottom contain components with high buoyant densities. Those components closer to the top of the tube have low buoyant densities.
That last part, guys, it's almost like making a suspension. So you're taking your whatever you're putting in it, in the glucose solution, and wherever it ends up, you know the density pretty much of it based on the density of that glucose. So kind of a cool way of doing it. Centrifuges are amazing, guys. If you've ever seen someone like get blood, like sometimes they'll do this after they take your blood if they don't need all of it, and usually it is in the lab, guys. Just most of the time you're not going to see it, but what they'll do is they'll take your blood and put it in a centrifuge, and that will kind of separate your erythrocytes, your red blood cells, from the plasma, the water-based part of your blood. Okay, so that's it for section 1, guys. That was more of an overview and just to give you a little bit of information about microscopes. And then we'll start relating the different cells to microscopes so you guys can see what we can see, what we can't see, that kind of thing. So we already went over this with respect to our domains. We talked about the bacteria, archaea, and eukarya. And then we talked about in eukarya, we have protists, fungi, animals, and plants. We talked about all that stuff. All right. In our eukarya domain, these are going to be our eukaryotic cells. Obviously, it's eukarya. The other domains, bacteria and archaea, these are going to be our prokaryotic cells. So basic parts of the cell, guys, here's 4 of them. Our cell membrane, that's the first one. Plasma membrane, another way to say it. We got our cytosol and our cytoplasm. So let me explain to you the difference between cytosol and cytoplasm. So if we have our, let me draw it out for you a little bit here. So we're going to have our cell. Okay, so here is our cell membrane. Okay. Here is our nucleus. Right. The cytoplasm is everything from here to here. That's our cytoplasm. Okay. Sorry about my M there. What is it? What's in there? What is in the cytoplasm? Well, there might be some mitochondria in there. Okay. There might be some little ribosomes around. Okay. There might be a Golgi apparatus somewhere. All right. All these different organelles. So the cytoplasm is going to be the area between our cell membrane and the nucleus. So it's the area between the cell membrane and the nucleus. Whatever's in there is part of the cytoplasm. Okay? How is this different than the cytosol? Well, go back to 7th grade or wherever when you first learned about cytoplasm and your teacher said, sound effect needed here, it is the inside of the cell, pretty much like this gelatinous material. Well, yeah, it kind of is. But that's really the cytosol. The cytosol is more like this gel-like material. Okay? If we wanted to draw the cytosol, it's all of the goop that holds all of our organelles into place. All right? The cytoplasm is going to be the goop plus our organelles. So there's your difference between them. We're also going to have some chromosomes in there. We're going to talk about the chromosomes later on. Those are going to carry all our genes. The chromosomes are going to be visible whenever the cell is reproducing. And then we already talked about the ribosomes in the past, and they make proteins. All right, so just going over the difference, guys. We have prokaryotic cells, no nucleus, no organelles. They just have ribosomes. And then they also have this area, instead of having a nucleus, it's called a nucleoid. It's just an area where they keep their DNA. Nothing too crazy there. And they still do have like a cytoplasm or a cytosol. Here is your pictures of the regular prokaryotic cell on the left, and then one under using a transmission electron microscope, okay? So, yeah, we could see all the inside stuff. Here's the nucleoid, so this would be our DNA up in here in the nucleoid region, okay? The nucleoid isn't really a fixed, nucleus, just so you guys know. It's just like an area, that's all. All right. Okay, difference between them and eukaryotic cells. Eukaryotic cells, you guys know, have a nucleus, have a nuclear envelope, okay? They also have the cytoplasm, just like the prokaryotic cells. Difference between them, though, in this cytoplasm, we're going to have all those organelles in it. Okay, talking about the cell membrane a little bit, guys, and then I think we're going to go into the math with the cell membrane. We'll go into that tomorrow, but just to give you a basic overview, it's the outside of the cell. It's going to allow things in and out of the cell. Okay, so with respect to that, guys, we need things inside our cell, and we need to take things out of the cell. Our cell's making proteins. Every cell in your body is doing this right now. Those proteins might be used by that cell, or they might be transported out of the cell to be used by maybe another cell. Okay. We also need oxygen. Oxygen is going to allow us to make energy inside of our cells in the mitochondria. So we always need oxygen, and we're always going to be bringing oxygen in. And that's able to go right through the cell membrane. Okay. Here is a picture of the cell membrane, guys. These purple things here, these are proteins embedded into our cell membrane. There's the phospholipids we talked about last chapter with the hydrophilic heads, which are the phosphates, and then the hydrophobic tails, which are the fatty acid chains. But we also have these proteins embedded in. And if you look at the end of the proteins, we're going to have these things called carbohydrate side chains, or we can also call them carbohydrate tags. None of this stuff, guys, has eyes. None of it can see where it's going. What these carbohydrate side chains do is they allow for certain molecules to pass through the proteins. So what's going to happen is, let's say we have glucose coming in. We got some nice sugar coming in. Sugar is not going to be able to go through each one of these proteins. The sugar actually has to find one of these tags that fits for glucose. And then once it finds that tag, it's going to be able to pass through. So let's say this one on the left here. Let's say this one. Let's say that one's for fructose. Well, if we have glucose coming and it goes to this one, oh no, you're for fructose. I'm sorry. Let me go away here. And then this one might be for glucose. So it'll find that one and it'll be like, oh, look, I could pass through, So that's how it works, guys. They're very specific, these proteins, to what they allow to pass through. Okay, we're going to start here tomorrow, guys. I want you to have a fresh mind for this one because there's some math involved. And we're also going to do a lab on this slide. So have a good rest of your day, guys.